Hello and welcome everyone to today's live class session of week 6. And today I will be discussing on the topic which was taught to you in week 6 lecture, that is enzyme classification and mechanisms related to it, and also discussing with you a few questions which will help you in solving the assignment. So I welcome you all. So welcome to the course on fundamentals of protein chemistry, which is taught by Professor Shagata Dasgupta of IIT Kharagpur. And so I will be just sharing the slides and discussing with you a few questions as well. Okay. So actually there was a few technical error, that's why there was a problem. Okay, good evening. So I guess my screen is visible to you.
Yes, sir.
Okay, that's good. So we can start. Okay, so in today's lecture, so previously we talked about proteins and the different ways of isolating proteins and how proteins can be, what proteins are made up of, the amino acid structure and all. So we have talked about proteins before. So today we will be not talking about proteins. We will be jumping into you can call a thing to be a protein. So that is your enzymes. So in today's lecture, we would be discussing about the primary or the basics of enzymes. Mostly it will be focusing, we will be focusing on the classification and the mechanism of the enzymes, and then on in the next class or in the next week we would be discussing about the kinetics, or which we call as the enzyme kinetics. So let us understand about the enzymes. So enzymes are basically proteins that increase the rate of a reaction by lowering the energy of activation. So enzymes are nothing but they are a kind of proteins that lowers the activation energy, thereby increasing the rate of the reaction. So basically what they do is they do not take part in a reaction, but they lower the activation energy, thereby increasing the rate of the reaction. But they do not primarily, or they do not actually take part in the reaction or react with your substrates. So therefore they are not altered or consumed during any reaction, and they are also reusable. So now we can categorize these enzymes into two parts. One is your the biologic catalyst, and then we have some chemical catalysts. So we also have some chemical catalysts, which are basically your, there can be some metal ions, or they can be some kind of salts or some reagents which you provide to increase the rate of a reaction. But we need to keep one thing in mind, that these biologic catalysts, so biologic catalysts are basically our enzymes, which are a kind of proteins. So what does these biologic catalysts do? So they have a few properties which are common between them and the chemical catalysts. So among them we have that these catalysts, either they may be your enzymes, or might be your chemical catalysts. They do not take part in the reaction, so therefore they are neither consumed or produced during a. Okay, so what does these enzymes have in common with the chemical catalyst? So mostly these includes your you can say that they never take part in a particular reaction, so neither they are consumed nor produced during the course of a reaction. Also these enzymes do not cause any reactions to take place, that is they do not react with your substrate and enhance the reaction. Rather their main task is to reduce the activation energy level, or that bump in the energy graph. So it reduces it and thereby enhances the reaction. But however these enzymes are also different from your chemical catalysts. So enzymes are basically your proteins, and these enzymes are highly specific and produce only expected products from the given reactants or the substrates. There are no side reactions which are actually taking place, which might occur in the case of your chemical catalyst, even though they do not cause any reaction. And these enzymes show high specificity, that is they know a particular sequence where they bind, and they are reducing the activation energy towards one substrate, or a broader specificity using more than one substrate. And these enzymes usually functions within a moderate pH and a temperature range. So these are just a understanding of how these enzymes or the biologic catalysts are different or similar to the chemical catalysts. Now this is an example of the lowering of the activation energy, where you see a frog where we have the rocks, and here it is a normal reaction without any enzyme. So it has a high energy barrier which you can see. So it only few number of frogs can only cross. So this crossing is termed as is between this reactants and the product. So there is an equilibrium, and the reaction can be termed as a slow process. Now when there is the presence of an enzyme, this frog the energy barrier is reduced, and therefore the frogs number of frogs jumping from the reactant to the product, or the number of moles of product formation from the particular reactant increases in one way. So this is a very nice cartoon diagram which will help you in understanding what is the actual role of enzyme. So there is an equilibrium between the reactant and the product, and the enzyme is responsible for shifting that equilibrium from the reactant towards the product side. Now the concept of free energy is that more free energy, less stable, greater work capacity. And you would be seeing that free energy decreases, so the direction of spontaneous change when delta G is less than 0 or has a negative value, the reaction is much more spontaneous. That is, imagine that it has a, from the reactant from A to B, the del G is negative, so the number of product formation or the ability of product formation in one way increases in that manner. So less free energy means more stable and less work capacity. So a thing that has lesser free energy will be more stable, and less free energy means in the negative value. So a greater negative value means less free energy, and therefore it is more stable. Now, in other way, you can also tell that when the free energy of the reactant is greater than that of the product, the free energy changes for the reaction is also negative. So the reaction proceeds spontaneously. That it has been shown in A. Whereas when the free energy of the product is greater than that of the reactant, then the free energy change of the reaction is positive and the reaction does not occur spontaneously. Therefore, the reverse reaction is much more feasible. So the percentage of molecules in the reactant site is more than that for the products, whereas when the delta G is negative, then the number of moles of reactants in the number of moles of reactants is basically less than the product, because the equilibrium is more shifted towards the product side, and in this case the equilibrium is shifted towards the reactant side. Now, there are the different components of an enzyme. Basically, we have the apoenzyme, which is mostly your protein portion, and which is mostly the protein, and that is basically inactive form. Okay, inactive form of the protein. Now, now we need something to actually activate it. So the activator or the non-protein portion is called the cofactor or the coenzyme. So we have an apoenzyme, which is the protein portion, and that is an inactive part of the enzyme. And we have a few cofactors, can be metal ions, can be some substrates and all, can be different non-protein portions that actually activates this entire system. And this entire system is known as the holoenzyme, that is the whole enzyme, which is the active form, and this active form comprises of your apoenzyme, and the coenzyme ultimately enables the binding of your substrate. So how many types of cofactors are there? So these can be your essential ions or coenzymes. So basically essential ions includes your metal ions, can be your divalent ions like magnesium, calcium, etc. And these are very tightly bound. You have some few activator ions that are responsible for activating your particular enzyme. So these are loosely bound, and on the other hand you have your coenzymes like your co-substrates and the prosthetic groups that are involved in the activation of your apoenzyme and ultimately binding of the substrate after it gets activated at one point of time. Now we have been speaking of active sites and what does the substrate go and bind. So we were speaking that the substrate goes and binds to the enzyme. It cannot go and bind anywhere. So the area on the enzyme where the substrate attaches or binds to the particular protein portion, that is called the active site of that particular enzyme. So imagine this is your enzyme and this is the binding site. This is your substrate. So this is where the catalytic site where your reaction is going to occur, and this is that also the binding site where your substrate is going to bind. So your substrate goes and binds over here. So as of now this is your coenzyme, or you can tell your holoenzyme. Okay, so something comes and activates it, so we are imagining it to be the active form, and then the substrate goes and binds over here, and this is the site where the reaction as well as the binding is taking place. Okay, now this is your actual enzyme, and the substrate is coming from the top and binding at the active site. Now the enzyme somewhat changes the shape to actually enhance the binding ability, because studied that it is more specific, more selective, and it is selective or specific over a broad range of space. So therefore this enzyme substrate complex has to be more stable or more tight. So for that the enzyme changes its shape or structure. After all, it's a protein molecule, so the proteins have the ability to rotate the bonds and attain different kinds of structures. So these attaining of different structures enables the binding of the particular substrate, and when this complex is formed, there are the enzyme and the products, and the enzyme moves out and the product is ultimately received. So we have the substrate which goes and binds to the enzyme. So substrate plus enzyme, and then we get the ES or the enzyme substrate complex. From there we get the EP, that is the enzyme product complex, and ultimately the enzyme and the product separate out. What we get is the enzyme as well as the product. So we isolate the product, and the enzyme can again be reused because it is not taking part in any kind of reaction. So there is a theory behind this enzyme kinetics, because we were speaking that the enzyme changes its shape and all. So how does it change shape, or why does it change shape? So there has been a few hypotheses and a few models or theories behind this. So the first one being your lock and key model. So what happens over here is that the place or the site where your substrate is going to bind to the enzyme is the active site, and in this active site it has a rigid structure. It cannot change shape, so it has a flexible conformation. So it has due to its flexible conformation, only those substrates which have that particular similar kind of a conformation can only go and bind to the active site, whereas other substrate cannot. So it is more specific, you can tell, or more selective, because only limited number of substrates can go and bind to this active site. However, because of this, a few other substrates, even though they have higher binding or tighter binding to your enzyme, might not be able to bind because of the lack of their respective structures. So this is one of the drawback of this theory, and this was the oldest model. So much more developed or a recently used proposed theory or model is the induced fit model. So what happens here is that the active site where the substrate is going to bind to the enzyme, here it has the ability to change its shape, and due to its changing shape it can enable or it can actually capture a lot many number of substrates. So it also increases the specificity, although it does not it is highly selective as well as it is more specific, and this model is much more consistent. So you see over here that it is the active site which is given a shape of a, say, a hexagon or something, but you have rhombus type structure of the substrate. So the enzyme attains a similar kind of a structure, and then the enzyme substrate complex is formed. For whereas what we looked into the lock and key model, it has a very rigid structure, thereby giving providing us a few drawbacks of the binding affinities and all. But this model is a much more developed model, and the active site being able to attain a lot many number of structures actually helps in capturing a greater number of substrate, and the shape of the enzyme active site and the substrate adjust to maximize the fit, which improves also the catalysis, and this also helps in the lowering the activation energy and enhancing the reaction process. Okay, now how do we classify these enzymes and how do we name them? So in chemistry we have studied about propane, ethane, we have started naming them, and this is what naming of enzymes, or we can call as the nomenclature of enzymes. So the IUB nomenclature of enzymes was developed in 1956 to create a systemic basis of the enzyme nomenclature, and this was a 4-digit number code where the first digit was the major class, the second digit was your subclass, and the third digit was your subclass, and the final digit was your specific enzyme type. Okay, so on the basis of this, it is a 4-digit code, and on the basis of the numbering process, the enzymes could have been classified. So one example of this is 2.7.1, and if you see over here, That 2 is your class name, that is your transferase. It belongs to the transferase type of an enzyme. So it is an ATP glucose phosphotransferase. So the transferase falls in the category of 2. 7 is the subclass name, that is its phosphotransferase, and 1 is your subclass, that is the hydroxyl group as acceptor, and the specific enzyme is your D-glucose as the phosphoryl group acceptor. So we need not keep in mind the name of the enzymes or the nomenclature in which, but if someone asks that it is a 4-digit code, so what are the 4 digits that they represent? So then we must be able to tell that the first one is the class name, that is, for example, it can be transferase, it can be oxidoreductase, hydrolases. Then the second one is the subclass name, that it can be phosphotransferase, methyltransferase, etc. Then we have the subclass, that is the third digit, which can be a hydroxyl group, or it can be an acetyl group, it can be a methyl group, it can be some other anions as well, or might be if it has the ability to capture a cation and all that. And the last one is the specific enzyme, that is D-glucose as the phosphoryl group acceptor. So this is what we need to keep in mind, and this is what will help us in having an approximate idea about the classification of these enzymes. And on the basis of this classification, a numerous number of enzymes have been developed till date, and mostly focusing on the class name or the type of enzymes, we have 7 differentiated classification of the enzyme. So one is the oxidoreductases, that is, it is basically involved in the redox reaction. Then transferases in different transfers and all, like phosphotransferases, that is the transfer of the PO4 3 minus group, that's phosphate group. Then we have the hydrolases, the lysases, then we have the isomerases, the ligases, and the translocases. So these are the numbering of the, these are the class name of the different kinds of enzymes. A very interesting or a very important term over here. So a very important term over here is catalytic efficiency. So the most enzyme catalyzed reaction are highly efficient, proceeding from 10 to the power 3 to 10 to the power 8 times faster than the uncatalyzed reaction. And so typically the enzyme molecule is capable of transforming 100 to 1000 substrate molecules into the product. So from the catalytic efficiency, the most important term being your turnover number. So what is this turnover number? So this turnover number is the number of molecules of substrate that is converted to your product per enzyme molecule per second. So imagine you have 100 enzymes, and you are measuring the turnover number for say 10 seconds. So for one particular enzyme, the ability of that particular enzyme or one molecule of that enzyme to transfer or to convert your product, your substrate to a product in 1 second is your turnover number. So if it has the ability to transfer two molecules of reactant to 2 molecules of product, so it has a turnover number of 2, okay, per second per molecule per enzyme. So that is your turnover number, and this is very important for understanding how good your enzyme is working, and or how specific or your enzyme is, how active your enzyme is, or how what is the activity of your enzyme. Another important term, activity of the enzyme, that can also be determined by this turnover number. Okay, so on the basis of classification, we have the oxidoreductases, the transfer of reducing equivalents from one redox system to another. Example, your alcohol dehydrogenase, the lactate dehydrogenase, and the cytochrome oxidase. The next one being your transferase, so it involves the functional group is transferred from one compound to another. Then you have among which you have the kinases, the transaminase, the phosphorilases, etc. So next you have the hydrolases. So these you can keep in mind, not mostly important, but yes, definitely the function of these enzymes are very, very important. You have to keep these things in mind about the enzymes, as to how these enzymes function and in what manner these enzymes function. You have the hydrolases, which cleaves the say your CO bond or the CN bond, the CS bond, the PO bond, by adding water across the bond. So water acts as the base or the nucleophile over here, and this actually is important for cleaving of those particular bonds. So among that you have the lipases, the acid phosphatases, which is important in your digestive process. The fourth one being your lyase, which cleaves the COCN-CS bonds, but they do so without adding of water. So in the previous step, the water was being added, which was actually acting as the nucleophile, or it was actually oxidizing or reducing the substrates. But here, the water molecule is not acting as the nucleophile, or we are not using any kind of water. So there are specific kind of these enzymes can function on their own and cleave the bonds in between these particular atoms. Okay, and example of these lyases includes your aldolase, fumarase, carbonic anhydrase, and there are many more kinds of these specific types. Another important or a very interesting type is your isomerase, that converts your particular molecule structure into another one, say keto and alcohol. Okay, so keto, we have the all structure, that is the alcohol structure. So like the isomerism is occurring, so they catalyze your intermolecular rearrangement of the functional groups, that again is in an equilibrium between the two particular conformations, and they can interconvert among themselves, which are basically your optical or your geometric isomers. Okay. So among these isomerases, we have the triose isomerase, we have the phosphohexose isomerase, we have the mutase that actually mutates something or changes the conformation. Okay. Then we have the ligase. So what is the function of the ligase? So it is actually forms a covalent bond between the substrate after utilizing the ATP or the ADP interconversion. So we have the DNA ligase, the glutamine synthetase. So what they do, what is the function of a ligase? It actually abstracts the ATP over here, and like you have the say what kind of a ligase you have, say a particular ligase which has, so these ligases have a very conserved lysine residue, and these lysine residue, so say when you are performing a ligation reaction and you have in the two ends you have a open OH and a 5 prime phosphate group. So what happens? So these lysine, because these ligases are enzymes, so they have particular conserved amino acidic residue. So these amino acids, mostly say lysine actually captures that ATP molecule and goes and binds to the 5 prime phosphate. And now these oxygen making now this becomes a very good living group. So the 3 prime OH, the oxygen over there actually goes and attacks to the phosphate group, and the group is actually becomes, as it is a better living group, so the living process becomes much more feasible and the ligation process is enhanced. So therefore this something or the other, by making it a better living group or by weakening the bond, by providing the water as a nucleophile and all, or changing, performing a mutation or changing a few conformation, so this will actually help in your a performing of different reactions. Okay, so in this manner or the other, the enzymes are enhancing your reaction process. Okay. Now we have a few environmental factors on which your enzymes or your say the functioning of the enzyme activity depends. One is your temperature. So this is your optimum temperature where your proteins works the most or is best over has better activity over here. So mostly it is 37 degree centigrade. There are different pH levels in which your proteins works the best. So like for pepsin it is around 1.8. For human amylase it is mostly at a neutral pH of 7.1, 7.2. Then you have trypsins which activates the most at around 8 point, uh, 8.4 to 8.5, and there are different ranges of pH in which the activity of the enzyme is enhanced. So in this way it actually increases your enzyme activity, and different enzymes works in different pH, so that is not much of an issue because um, it does not actually enhances the activity of the reaction because these enzymes are anyway not taking part in the reaction. So the pH and the temperature becomes much more effective. But if you are putting a enzyme which shows greater activity in neutral pH and you put it in a acidic environment, the activity actually stops or is ceased. The enzyme might get actually affected and the protein might become non-functional. Okay, so understanding the pH and the temperature of the enzymes is very, very critical. And then we come to the substrate concentration and the reactant rate of these enzymes. So we will be talking about these enzyme kinetics in details in our later classes, okay, or in the next class probably. So in short, if we talk, the rate of the reaction increases as the substrate concentration increases, but why we would be coming to the derivation and all in the next class. At a constant enzyme concentration, and the rate of the reaction increases with the substrate concentration. And you can see here from the graph where we have the initial reaction rate and here we have the substrate concentration. So the maximum activity is at 10, and the maximum activity occurs when the enzyme is saturated. So when all the enzymes are the binding substrate, so at that point you observe the maximum activity of a enzyme. So when all the protein molecules are bound to the substrate, or when all the enzymes are bound to a substrate and there can be no other vacant space in the enzyme, at that time you would be observing the maximum activity of the enzyme, or that we call as the maximum velocity. So here we see that it is only less, only few sites are bound. Here mostly are bound, and now it reaches a saturation level where no more sites are free for binding. Okay. So previously we had talked about the catalytic power. Once more I am repeating the scientific definition of it. So catalytic power is defined as the ratio of the enzyme catalyzed rate of the reaction to the uncatalyzed rate. So for example, urease, so at 20 degrees centigrade, the rate constant of the enzyme catalyzed reaction is 3 into 10 to the power 4 per second, whereas for the uncatalyzed reaction, it is 3 into 10 to the power 10. So when we perform the ratio of it, then we get it to be, I think there is an error. Okay, so this is not 10 to the power 14. So ratio of the enzyme catalyzed rate to the uncatalyzed. So the catalyzed rate is 3 into 10 to the power 4, and this is your 10 to the power 10. So 4 by 10 to the power 10, that is 10 to the power minus 6 per second. Okay, so that is going to be your catalytic power. Now another important term is specificity. So defined as the specificity of the enzymes for the reactants upon which they act. So in an enzyme catalyzed reaction, none of the substrate is diverted into non-productive side reaction. So no wasteful byproducts are actually produced over here. Okay. And the substrate upon which an enzyme acts is called the substrate. The selective qualities of an enzyme are collectively recognized, and it is termed as your specificity. And the specific site on the enzyme where the substrate binds and catalysis occurs is called your active site. So these three are very important terms, and apart from that, the turnover number, the catalytic power, these are also very important. And mostly this equation, because there might be a few questions on one or two question on this, so just for safety we need to keep these things in mind. Now coming to the regulation of these enzymes. Now the regulation of enzyme activity ensures that the rate of the metabolic reactions is appropriate to the cellular requirements, because only then the regulation process is complete, because an enzyme can react in any manner, but it does not matter if it is not functional inside the body. So we try mostly in vitro or in vivo, that is different, but inside the body how it acts, that is mostly very important. So it is essential to the integration and the regulation of the metabolism. So there are a few enzymes that act as your inhibitors, activators. There can be regulates the rate of synthesis. They have different hormonal activities. So there are different kinds of enzymes and they function very differently depending on in which category they fall and all. Okay, so that is okay, fine with that. But the regulation process is very important, because after all these enzymes are going to be put inside your body, and how they are going to function over there. For that, in vivo studies are performed to understand the regulation of it. The structure can be understood from in vitro experiments, how they are binding and all, about the specificity, the selectivity, that is all about the in vitro experiments and all, that is outside the body. But how it is going to function inside the body, that is much more critical. So specificity is highly specific compared to other catalysts. It catalyzes only the specific reactions, and there are three types of specificity. One is your specificity, optical specificity. That is, it is specific to some kind of isomers and all, or some kinds of optically active molecules and all. Then it is reaction specific, and it can be substrate specific as well. So the reaction specific means it performs this kind of reaction. So like for transferase enzyme, if you put for ligation, it will never perform. And so that is, it has to be put in that particular reaction that you're wanting to actually perform. A transferase won't act in a ligation reaction, or a ligase won't be act in a transferring reaction. So that is what is about reaction specificity. And another thing is the substrate specificity, that is also very critical, and it is binding to a particular kind of a substrate. It does not bind to any substrate, because it has a particular recognition or identification site on which it goes and binds. So therefore enzymes are very highly specific. So here is the optical selectivity, which is able to recognize only your optical isomers of the substrate, and it acts only on one type of isomer. So for example, you have two enantiomers, but it is going to bind to, say, your D form or your L form. So that is that. So enzymes of amino acid metabolism, DNA amino acid oxidase. So isomerase do not exhibit stereospecificity, because your isomerase is involved in the conversion from one particular isomer to another isomer. So this is the only enzyme that does not exhibit stereospecificity. Okay, now coming to the reaction specificity, which catalyzes only one type of specific reaction over the substrate. Your amino acids can undergo deamination, transamination, then you have the decarboxylation, and each of them is catalyzed by a separate kind of a enzyme. So that is your reaction specificity. And finally, a substrate specificity, that is specific towards their substrate. Like you have the absolute substrate specificity, relative specificity, and the broad substrate specificity. And the example of the substrate specificity is your glucokinase, galactokinase. So both transfer phosphoryl group from the ATP to the different molecules. So what is this absolute substrate specificity? So they act on only one substrate, like your urease that acts on urea. Now the relative substrate specificity, it can act on structurally related substrate. So if two molecules have a similar kind of a structure, or might be they function differently, but the structure or the amino acid sequence where your enzyme is going to go and bind, if that is same, then it is termed as your relative substrate specificity. And these can be further divided into your group dependent and bond specificity. So the group dependent relative specificity is it acts on a specific group. Example, your trypsin, which breaks peptide bonds between lysine and arginine. You have chymotrypsin that acts on aromatic amino acids. And bond specificity is your glycosidases and your proteolytic enzyme that cleaves after a few, that cleaves the particular bond. So they are acting on the specific kinds of a bond. So this is very, very, very important slide, because there will be a few questions on a few enzymes like trypsin and chymotrypsin, and there are others also where you have cyanobromide. Okay, that's a chemical that breaks the bonds to the C terminus of your methionine, and there are other enzymes. You can study them, or you will get them in the broader lecture of this particular week. Okay, so what does trypsin do? It breaks the peptide bonds between lysine and arginine, and chymotrypsin acts on aromatic amino acids. And then you have the bond specificity and all. Okay, so what is so we spoke about absolute specificity, we spoke about relative specificity, and now coming to the broad specificity. So what is broad specificity? It acts on closely related substrate, like you have hexokinase that acts on different hexoses. Okay, so there are different kinds of hexoses. There can be different kinds of pentoses and trioses and all. So on the similar closely related substrates and all, they are actually acting. So therefore they have a broader specificity. And if we speak on the chemical nature and the properties of enzymes, these enzymes can be your proteins, and sometimes they can be RNA. So their tertiary structure and specific conformation, that is actually essential for the function. Like we studied that a folded protein has greater functions compared to an unfolded or a denatured one. So therefore enzymes, being in the category of your proteins, have highly have tertiary or structures, and they have a specific conformation which actually enhances their catalytic activity and their catalytic power. And then they have a holoenzyme, that is the functional unit which combines of your apoenzyme and the coenzyme. So what is the difference between a prosthetic group and a coenzyme or a cofactor? So prosthetic group is a because this slide is very, very important. So go through this slide at least twice, because there will be questions on these two, and the difference is very, very important, where you have the prosthetic group, which is the non-protein portion, and the cofactor or the coenzyme is the non-protein molecule. Okay, and then you have the prosthetic group, which tightly covalently binds, whereas your cofactor binds loosely. Then you have the prosthetic group that is stable incorporation, but your coenzyme is dissociable. Your prosthetic group cannot be dissociated, but your coenzyme can be separated by dialysis, because your coenzymes or your cofactors are very, very small molecules, and in the cutoff range they do not fall above the cutoff or the threshold value of the dialysis bag, because the dialysis bag have very small pores that do not let your protein move out, but it actually maintains an equilibrium between the solution and the protein inside the dialysis bag. So there is an equilibrium, or there is an osmosis process that is going across. So but the cofactors, being very, very small molecules, they do not actually stay inside the dialysis bag, hence can be separated by dialysis. But these prosthetic groups, being the non-protein molecules, even though they are non-protein molecules, they are bigger in size and they cannot be dissociated. Okay, now coming to the problems. Okay, so you people tell me the answers, and we can be discussing them now. So that was all about the lecture and all. Nothing great is over here, but we need to have a better idea about the classification of the enzymes. And from here you can actually expect direct questions more than the indirect ones. So these are going to be quite easy questions. that you can actually understand after I am solving with you the means the question and answers. Okay, so I think we can begin. Just a minute. So we have only one student. Okay, no problem. Okay. So let us see what is the first answer to this question. The site where the catalytic activity of an enzyme generally takes place. What is this site called? So what do we call this site? No answer. Okay. So we call this site as the active site. Okay. The next question. Given that del G for the reaction is negative in the reaction equilibrium favors the formation of which substance? So which of the substance is going to form in this case if the delta G naught for the reaction is negative in your S2P direction? You have a substrate, you have a product, which substance is going to form? So Kavivarthi, are you there? Okay, I think you are there. So try answering the question. Never mind if it's wrong or if it's incorrect. Good. So try answering. That will also help you enhance your interaction skills, as well as it will also help you in answering questions in your later stage, like in solving assignments and in the exams. And these are easy questions and direct questions, you can answer them. So delta G naught is negative, and that means the reaction is feasible enough. So from substrate it is going to the product. So what substance you will get in more amount if your delta G naught is negative? That is what it asks. So if you see that initially P and later S, no, that is not correct. It will be basically P, because your reaction is very feasible. Okay, so it is negative, means your reaction is feasible enough. Okay, and what does feasibility means? That it will help in the, and the direction is also given, substrate to product. So in this direction the reaction is feasible, that the product will be forming in more amount. So product will be in more concentration if delta G is negative in this direction, S2P. Okay, never mind. So for the next one, for the reaction is plus E to ES to E plus P, where you have the substrate, enzyme and the product. The quantities can be measured experimentally. So which of the following can you measure experimentally? Is it the substrate, the product, the total enzyme, or the bound enzyme, or the bound product, or the bound substrate, or the free product? What can you measure from here? So see, we have the substrate that we can measure. Substrate and product, okay, that is correct. I won't say that is incorrect, that is true. But only substrate and product, no, there in that place it is incorrect. Substrate and product is correct because we know the concentration, we know it, but we also know the total enzyme that we are providing in the beginning. So all the beginning concentrations are known to us. Okay, so we can understand the substrate, the product, as well as the total enzyme, because how much is utilized, that we cannot know, because the product, this we cannot find out. Okay, but apart from the bound fractions, everything else we can get. So therefore A is the correct option. I think you're getting my explanations well. So apart from the bound one, because this is an intermediate step, we cannot get the concentration, but apart from that, all others are measurable. Okay, the next question. Which of the following determines substrate specificity of an enzyme? So what determines the substrate specificity of an enzyme? So what is substrate specificity of an enzyme determined by? I just discussed about them. So if you see that what are the options given, the complementary geometry of the substrate and the enzyme, determining the substrate specificity of an enzyme. So the complementary geometry of the substrate and enzyme is very important, because it has to be having a complementary structure, be it via the lock and key model or the induced fit model. The geometry is very important, and the complementary electrostatics of the substrate and the enzyme, that is also very important, because the bond that is going to form, or the interaction that is going to occur between the substrate and the enzyme, that is also very critical. So both your option A and B would be the correct answer over here. Are you getting my explanations well? If you have any problem, you can ask me. Okay, I guess my explanations are clear to you. Okay, moving on to the next question. While monitoring the activity of the enzyme in UV spectrometer, which two parameters are monitored in the instrument? So when we are um, studying the activity of the enzyme, what is there in the x-axis and what is there in the y-axis? Try to answer this one. At least try to give an answer with proper understanding, definitely. B, time versus wavelength. Okay, no, that is actually not the correct answer. See what we have over here: absorbance in x-axis and wavelength in y-axis. That is not correct, because we understand that we are providing a wavelength. That is true, because that is protein 280 nanometer, that is known to us. So what else? So we need to have absorbance. We need to have absorbance. So absorbance is important, but in which axis should it be absorbent? It should be in the y-axis. So the absorbance is always in the y-axis, because we are also measuring the intensity and all. So for that, absorbance is placed in the y-axis, and only one option we have absorbance in the y-axis, and in the x-axis we are going to have time. So D is the correct option. So here we have a graph. We have like this: we have absorbance and we have time, and then with this you might be having like say like this, you are getting a graph, and like this and all. Okay, so that will be your absorbance. Okay, the next one. A rate versus pH curve with an inflection point at pH 4 suggests the involvement of an dash in the catalytic step. So you are plotting a rate versus pH curve with an inflection point at pH 4. So what is there in the catalytic step? So the pH is 4. So what type of an amino acid or ion would you expect in the catalytic site? Very good. So you need to have an acidic amino acid. Yes, because the pH is in the acidic range. Correct. The next one. Choose the incorrect statement about the multi-step reaction. So which of the statements over here are incorrect? The free energy of the intermediate must be greater than the transition state. The transition state with the highest free energy always corresponds to the rate determining step. The free energy of the intermediate may be greater than that of the reactant, or by catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. So which of the options are incorrect over here? So what are the options given? 1, 2, 3, 2 and 4, 2 and 3, 1 and 4. Which of the following do you feel are incorrect? D. Okay, but see option number. Okay, let us see from the first option. The free energy of the intermediate must be greater than the transition state. That is incorrect. It cannot be greater than the transition state, then the reaction will not occur. So 1. So let us see in which we have 1. So in A and in D. Okay. Okay, coming to the next one. The transition state with the highest free energy always corresponds to the rate determining step. Okay, so with the highest free energy always corresponds to the rate determining step. That is also incorrect. And number 3, it says the free energy of the intermediate may be greater than the reactant. So it is said may be greater than the reactant, but mostly it is greater than the reactant. So option 3 is also incorrect. So okay, so okay, no, wait. Okay, so there has been a error. Okay, so the free energy of the intermediate must be greater than the transition state. That is okay, so it has been asked what is the incorrect statement. Okay, the transition state with the highest free energy corresponds to the rate determining step. That is correct. Yes, the free energy of the intermediate may be greater than that of the reactant. The free energy of the intermediate is greater than that of the reactant. That is also correct. By catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. Yes, that is incorrect. Very good. So option D is the right answer. Very good. Okay. The next one. Methionine synthase catalyzes the conversion of homocysteine into methionine in the presence of vitamin B12. So here vitamin B12 plays the role of an apoenzyme, holoenzyme, cofactor, prosthetic group. So methionine synthase is catalyzing your homocysteine to methionine conversion in presence of B12. So what is B12 actually doing over here? So apoenzyme or holoenzyme. Okay, no, we have the enzyme over here. So A and B is not correct. So neither this is the inactive form, or the B12 is neither the inactive form nor the active form. So A and B is not correct. So whether it is a cofactor or a prosthetic group, what is it? So basically B12 is a cofactor because it is playing the role, because it is activating or enhancing the conversion process. So option C is correct. The next one, choose the incorrect statement about the enzyme. Compared to an uncatalyzed reaction, the enzyme speeds up the rate at which a chemical reaction approaches equilibrium. Enzymes make the reaction thermodynamically favorable. Enzymes decrease delta G reaction as well as delta G hash. And in the presence of an enzyme, all thermodynamic parameters of the reaction are altered. So which of the options are incorrect over here? Okay, let us go through them one by one. Compared to an uncatalyzed reaction, an enzyme speeds up the rate at which the chemical reaction approaches equilibrium. This is the correct option. Okay. So one is, wherever one is there, it is not correct. So C option is gone. Let us see number 2. Enzymes make our reaction thermodynamically favorable. Enzyme is not taking part in the reaction. Whatever it is doing, it is not affecting any thermodynamic parameter. But what it is doing, it is actually lowering the activation energy. So it is directly related to the activation energy rather than the thermodynamic parameters. So as it is not affecting the thermodynamic parameters, therefore option 2 is not correct. And option 3 is also not correct. And in the presence of an enzyme, all thermodynamic parameters are altered. Therefore, this is also incorrect. So it is not affecting the thermodynamic parameters. That is very, very important. So because of that, it is not changing or altering the thermodynamic processes and all. Therefore, A is the correct option. The next one. The enzyme active site should preferably be analogous to. So what should the enzyme active site be analogous to? Is it the product, the substrate, or the transition state? With the substrate, what did we see over here? Just going back a few slides. See, here we have the substrate. We also have the substrate and the enzyme complex, and then this is also going to form the product, and this is your transition state. Because see, the substrate might bind, that is fine, but if the transition state or the complex is not formed, then how will the product be formed? That is another question that arises. So because of, for that, not only the substrate or the product has to be analogous, but also the transition state has to be more analogous to it. So because of that, the transition state, the product, as well as the substrate, so all three of them are correct, but we have to choose only one of them. So we can stick with 3. We can choose 3 of them also. So A, B, C, D, all of them are correct. So only option does not lie over here. So it has to be analogous to all of them. But if only you have to choose one of them, so it is 3. So keep this in mind. If it is an MSQ, go for 1, 2, 3. But if it is an MCQ, try going for 3, because the transition state is the intermediate state and it lies in between the substrate and the product. Okay, the next question. So this is a reaction that is being shown over here. So which enzyme or which group of enzyme performs this reaction? Is it hydrolase, ligase, oxidoreductase, or transferase? So what is this? What type of enzyme is involved in this reaction? So now this type of questions, to understand this type of or to answer this type of questions, you have to need you need to have a proper idea about what these enzymes or in which manner these enzymes are functioning. So what is over here? So you have NH2, you have a CO that is conserved over here, and now this NH2 is going and binding probably. So there is a transfer that is going on, okay, because this NH2 group, because this particular group is transferred over here, and this is a leaving group. So this goes, and this entire NH2 goes and binds over here, okay, it attacks over here, and then this group leaves. So ultimately you get this product. So this is performed by your transferase. Very good. Okay, the next one and the last question for today. So this is another reaction that is going on, and which enzyme or which group of enzyme is performing this reaction? So what is happening over here? Let us see. So there are a loss of few groups and a double bond is being formed. So what or which enzyme performs this reaction? And there is an equilibrium in between them. So this enzyme, so a hint is that this enzyme can perform both of these reactions. It is not a one-sided reaction. It can perform both the reactions. So which type of enzyme can do that? So in one way it is a double bond formation, and in the other way it is the double bond that is breaking, and it is the incorporation of a few functional groups. So what are the options over here? We have transferase, lyase, ligase, and the hydrolase. So what does transferase do? It performs a transfer. Here no transfer is occurring, so transferase is not correct. Uh, what about ligase? So here no ligation is also occurring. So we have hydrolase and lyase, but for hydrolase you need a water molecule to be acting as the nucleophile, and then it attacks over here or here, and then it is the bond is breaking or the joining is occurring. Okay, might be there is a loss of an OH group and the double bond is formed. But here is no water molecule, so hydrolase also is incorrect. And the other option that is left is lyase. So lyase is the correct option, because what is was the function of lyase? It actually acts as the bond breaker or a bond or the formation of a bond, okay, by without the help of any oxidizing or reducing agents, or without using any kind of water molecule. So therefore lyses are the right, therefore lyase is the right option. Okay, so that's all from the question and answers. I think that is all. So if you do not have any question, we can end the class. And you can get back to me in my mail as well. I am typing over here. I think you know my mail ID. So you can write back to me if you're having problems with the assignment and all. I will get back to you. So that's all then. Thank you.
Thank you, sir.